welcome everybody, to our EPI seminar. It's not as cold here in Birmingham, but I know in Northeast, there's a big storm. So hopefully, if you have friends, family there, they are all safe and warm. But even here, stay warm. So today we have Dr. Richard Reynolds. He's an associate professor in the Division of Immunology and Rheumatology in the Hersing School of Medicine here at UAB across campus. After his PhD in biology from University of Maryland, he joined the section on statistical genetics. Dr. Tiwari was one of the section heads of that, which was then in the Department of Biostatistics. So in that term, he's not a new face to School of Public Health. Then he joined the Division of Immunology and Rheumatology as an assistant professor in 2010. His research is focused on genetics and in some kind of omics studies of gout. So with that, Dr. Reynolds, welcome to the EPI seminar.
Okay, thank you, Sadiq. Thanks for inviting me. It really is kind of fun to be here for many reasons, but one of them is because I should say, I think it was it 16 or 18? I can't do the math. It was a long time ago. 18 years ago, I think. I was here in 2008 in February giving a talk to apply for the T32 postdoc that David Allison and Haymont were PIs of. And I actually went back to the slide deck of the presentation and pulled these pictures. So, I know, I can imagine what you're thinking. What does gout have to do with flowers and pollinators? But Sudeep had asked that I kind of give a little kind of history of my path to my current position, and it essentially is plants to gout, and so these are some pollinators of three closely related species of Silene. And so I did my dissertation work on the evolution of pollination syndromes in plants. And these were the species that I studied. And I was really interested in kind of measuring phenotypic selection on floral traits in these species. And phenotypic selection sort of defined as within generation change in just an actual phenotype. And so I was interested in like petal length, corolla tube length, and things like that, and then relating the patterns of selection that we detected to the major pollinators of of the plants. And so it was a very kind of quantitative PhD, I would say, and I took a ton of statistics, and I was really interested in that. Here's some more pictures. It's sort of like catnip for plant lovers, I guess. This is the picture in the upper left is a small plant that's called fire pink, and this is in its natural environment. Where I studied it in southwestern Virginia for my PhD. But you can also find it if you're on a hike at, say, Ruffner Mountain. You'll see these flowering here in April, maybe May, and they're really quite common there at Ruffner. So, yes, really interested in statistics, and I was thinking, what do I do with my life? And, I wanted to get some more training in biostats. What led me to apply to the T32 program here at UAB. I took this slide as well as from that old slide deck, and it's quite illuminating, I think. So again, 18 years ago. So I was thinking this is what I've done, and this is kind of what I want to do. And what I felt like I wanted to do at 18 years ago was to do some more kind of methods development, and to kind of start to think about disease traits as multivariate phenotype. So lots of different traits that are correlated. Does that comprise a disease trait? Similar to like how I viewed adaptations, in my dissertation work, which were just pairs of correlated characters. And then, of course, I was interested in this new technology. You could use big gene chips and get lots of genotype data, and then look at associations with disease phenotypes. So that's kind of what I was thinking I wanted to do, and actually, it's pretty close to what I'm doing now, which is kind of fun to think about. But and of course, this was made possible by many mentors. A lot of many that are sitting in this room. It's fun to see familiar faces. I see Tony and Haymont and Ryan. So it's so glad y'all are here. This is just one of the kind of early methods development papers that I published, and this work came out of as a side project out of my postdoc here, but this actually made the blogosphere, and as the most difficult title of any scientific paper published in the journal Evolution. That's supposed to be a joke, but I probably should have thought twice about that. But now it's a permanent scientific literature. So anyway, let's kind of move on to to my current current interest. So I think that currently really interested in mechanisms of inflammation in crystal-mediated autoinflammatory diseases, particularly gout. For the last 10 years, really, I've been studying it, maybe eight to 10. And again, I think you can kind of summarize this as my sort of scientific career. It's gone from flowers to gout, which is a little bit weird to hear, but it's there actually are some similarities there. So let's I'm sure that there are a lot of people in here that are unfamiliar with gout and its characteristics, and I want to just kind of go through this, just some basic background on it. Gout is ultimately a response to monosodium urate crystals that precipitate out of blood and then sort of lodge in cartilage and joint tissue. The immune system absolutely hates the crystals, and the immune system gets signaled that the crystals are there through this NLRP3 inflammasome, and that's a really powerful innate immune system sort of molecular mechanism that generates cytokines that sort of attract inflammatory cells to the site of inflammation. The gout flares is sort of the I guess, how should I say it, just the sentinel clinical characteristic of gout is the acute gout flare. It's really painful. You don't want to have them. And how do you prevent them? Long-term urate lowering therapy is one way to do that. So monosodium urate crystals are the result of crystals dissolving out of solution of serum urate or uric acid. People sort of use those interchangeably, but the correct term is serum urate. Urate or uric acid is the end product of purine metabolism, and the enzyme that converts xanthine and hypoxanthine to uric acid is called xanthine oxidase, and so the urate lowering therapies are xanthine oxidase inhibitors. So they prevent the production of uric acid, and thereby lowering uric acid levels in the blood. So what triggers the flares? a lot of people say that, I had a steak or I drank too much beer or whatever, and then that, that's a trigger for my gout flare. But actually, most people don't point to a trigger, and it's just sort of almost an episodic, sort of random thing that occurs, but it gets your attention. And ultimately, you could have what's called chronic tophaceous gout, and that sort of the most severe form of gout would be clinicians can palpate tophi that are in your joints, and the tophi are sort of these. Is this point? I'm like pointing to this, but maybe can y'all see a cursor when I do this? You can't, can you? I wonder how to do that. Does it just go over? No. We don't need a pointer. So in the bottom right here is a picture of the tophus. So it's just a collection of crystals and inflammatory cells that sort of surround it, and it's almost, people describe it as a granuloma, but they're both innate and adaptive immune cells that are associated with this, and the goal is to dissolve these tophi eventually. Gout is a fairly significant public health problem, so its prevalence is 4% or so of adults. It's more common in males than females. Females don't really get it until their age they're at menopause or older, and this graph just sort of shows why this has our attention, and this is a graph of hospitalizations in gout versus rheumatoid arthritis over a 15-20 year period. And you can see that RA hospitalizations are declining while gout is increasing. So, this is something that's worthy, I think, of study. So this graph is a phylogeny, and this would be something that evolutionary biologists would look at and like kind of get and understand. But what we have here is basically a gene phylogeny of the sequence, the genetic sequence of urate oxidase, which is also called the uricase gene. Uricase is the enzyme that breaks down uric acid to elantoin. But this is a phylogeny of the sequence of urate oxidase in higher primates and some lower primates, and you can see near the bottom, those primates have a functional urate oxidase gene, and the higher primates, including the hominoid taxa, have a non-functional urate oxidase gene. So ultimately, the reason that human beings get gout is because they do not metabolize uric acid. That's the end product of purine metabolism in humans. It's a really interesting story, and in fact, it kind of got my attention because, of course, with my background in evolutionary biology, but one of the things that these authors did was look at the ratio of synonymous to non-synonymous mutations in the exons of this gene, and they're shown above the bars. But what is clear here is that there is an increase in the number of non-synonymous mutations, which are amino acid causing changes, and in the taxa that have the non-functional urate oxidase gene. So what is that telling us? That's just telling us that because this gene is non-functional, selection is silent towards those those non-synonymous changes, and purifying selection is still operating in the taxa with a functional urate oxidase gene, but not in the taxa with a non-functional urate oxidase gene. So it turns out that there have been at least two, probably more, independent loss of function mutations in these these primates and the higher primates. It involves an arginine to a stop codon, and there's one that was in the ancestor of the hominoid taxa, and then a separate one in gibbons. But whenever you see that in evolution, where you have these two independent, two changes that occur in independent taxa, that we call that convergent evolution. And it suggests that natural selection is sort of behind that. And one of the sort of great mysteries is what was the source of selection that was driving the loss of function of urate oxidase. So the immediate phenotypic effect would be higher serum urate levels, and that's been measured. I mean, I think that in taxa with a non-functional urate oxidase gene, the urate levels are 10 times higher, at least, than taxa with a functional urate oxidase gene. So there you have it. That's kind of some little bit of background of why human beings get gout. It's a cool story. And I asked the clinicians, do they tell their patients this story? And they just kind of stare at me. No, I don't think they do. But when I tell my friends about it, they're like, "Oh my gosh, that's an incredible story." Not everybody knows that we don't have a functional urate oxidase gene. I'm not going to go through this slide too much, but unless Sudeep, do we have 2 hours? Just one. Okay. All right. So it turns out that loss of function of the urate oxidase gene was sort of gradual over the course of mammalian evolution in terms of the transport capacity of uric acid. So here's our first gene. You could put it in your pocket and take it home. You can talk to your friends at the cocktail parties. Urate oxidase gene. This is associated with gout. This is from Ref-seq, so it catalyzes the oxidation of uric acid to allantoin. This gene has been inactivated by mutation and is non-functional in humans and some other primates. So what I want to do now is talk a little bit about epidemiology, just to give you a sense for comorbidities of gout. So gout doesn't just kind of express itself in a vacuum. There are other things that are associated with it. And this is a paper that I was involved in. It was just a study in using the atherosclerosis risk in community study. We just looked at the relationship between various traits and incident gout in this cohort. What we found was that ethnicity and systolic blood pressure was associated with incident gout, significantly associated. Of course, along with serum urate. So you can see on this graph the bottom axis, so we call it the x-axis, has serum urate from 2 to 12, and you can kind of see like between 6 and 8, there's sort of a liftoff of probability of gout there. Well, that happens to be about the concentration of serum urate in the blood at which crystals start to form. So you can kind of see that spectacularly here. But what you also can see is that, say, if you have European ancestry and you have normal blood pressure, that your risk for gout is a good bit lower at a given level of serum urate. So I just thought that was that's a neat paper to be involved with, and kind of our first dip in the toe in epidemiology of gout. Here's another study. Lissandro, who's a great investigator here in Epi, but he did a study using the regards data of cardiovascular disease risk with people with gout. And for gout, there were about 2 times the hazard risk of heart failure than non-gout participant in regards, and the all-cause mortality was a good bit higher for participants with gout than the non-gout. So cardiovascular disease really does seem to be associated with gout as a comorbidity. This study kind of was really kind of interesting because it's kind of maybe got a little bit more to the mechanism. What's driving the increased risk of cardiovascular disease in people with gout? So this is a group from England, and they had a large registry of people in England, and were able to ascertain the time of gout flare, and then in temporal windows from that index date, we're able to then assess the risk of cardiovascular disease cases. And what they showed was that if you flared within 2 months, you had a much higher risk of a cardiovascular event than if the flare had occurred 4 to 6 months prior. So that kind of tells you something about the pathogenesis of gout-related cardiovascular disease. It may be associated with the acute flares, that massive inflammation that occurs when your immune system is getting angry at the crystals. So gout and hyperuricemia, again, like hyperuricemia being urate levels that are above the crystallization threshold, are associated with hypertension, cardiovascular disease, heart failure, and stroke. There are a number of other comorbidities that are also associated with gout, so chronic kidney disease, obesity, and diabetes are also associated. Chronic kidney disease is an interesting one, but probably the mechanism there has something to do with the fact that if you have really poor kidney function, you don't filter urate as well in the kidney. So that probably has something to do with it. But I think in my mind as an investigator, making this observation that gout and its sort of expresses itself with a number of different correlated phenotypes, comorbidities, and if I'm interested in genetics of these of gout and these comorbidities, how do you approach that problem? And so I as a first stab at this problem worked with Ryan and Anna Vasquez, who's at Michigan State now, and she and we were like, let's use some multivariate genetic models and estimate heritabilities and genetic correlations between these traits that are kind of pointing to these comorbidities, and see if we can learn anything about the genetics of all of these traits together. So that's kind of the next thing I want to talk about. It was published in the European Journal of Human Genetics. But we had two different data sets, thanks to Don Arnett and Ryan. We had the Hypergen data, that's the Hypertension Genetic Epidemiology Network study, and we also used the Framingham Heart Study. So what we had genetic markers from both of these, and both of these studies had somewhat of a family structure. I think there were, if you look at it, I mean, most of the individuals are unrelated, but there were some cousins and brothers and things like that, first degree and second degree relatives in the data set. So you can use genetic markers to compute like pairwise genetic similarities between the individuals in the study, and then use that matrix of genetic similarities to compute heritability. And that's what we did. The heritabilities were pretty close between Hypergen and Framingham Heart Study for the traits that we studied, which are from left to right: serum urate, serum creatinine, BMI, systolic blood pressure, and glucose. I'm going to show you some data on the genetic correlations next for Framingham Heart Study. This is kind of busy, but on the diagonal are the heritabilities for the different traits that I just showed you as a bar graph. And then on the off diagonal are the genetic correlations. And so those genetic correlations are really estimating the shared genetic component for the pairs of traits. And you can see here that they range from positive for like serum urate and serum creatinine. Serum creatinine is sort of our kidney function measure. The serum urate and BMI was positive. Now serum creatinine was negative for all of these traits, which was kind of interesting. It's had a little bit of different pattern. A lot of these are 95%. I think they're posterior credible intervals. So these anything that doesn't overlap 0 is worthwhile, significant. Here's hypergen. Very similar pattern. You get the positive genetic correlations between urate, BMI, and glucose, and then negative genetic correlations estimated with serum creatinine. So it was remarkable. We have two totally different independent data sets, and we're estimating these genetic parameters and from marker data, and they're very similar across the board. So that was kind of encouraging. That's what's shown here is the parameter estimates for Framingham versus hypergen. So this was like I would say it was really hard to interpret the results. As much as I wanted to do this study, it was like, how do we explain that? How do we kind of boil it down and summarize it? And I mean, the fact is that we have genetic correlations. We have shared genetic, effects on pairs of traits. That's important. So if it's a positive genetic correlation, then we say that gene positive deviations in that genetic effect is simultaneously affecting the two traits in the same direction. Same with the negative. If you have a negative genetic effect, it's going to lower the expression of the two traits. Or you could have a negative genetic correlation. So that's the opposite pattern, where you have a positive deviation results in a for one trait, it results in a negative deviation for the other trait. So those are kind of the two possibilities that you can have. Anyway, it was kind of challenging, but to just to kind of summarize. So I have here some plots. Basically, the top plot and the top row and the bottom row are the same. It's just that I've kind of highlighted the different traits. But what stood out to me and us, I think, is that it looked like we kind of had two independent or separate kind of axes of genetic variation, with urate and kind of comprising one mode or node of genetic variation, and then creatinine another that looked very different. And so it's sort of, that was one of the conclusions that we made in the study, that there are these independent kind of axes of genetic variation that are responsible for the trait expression of traits that are associated with gout and its comorbidities. So I mentioned that we're computing and estimating genetic correlations that give us information about the shared genetic basis of pairs of traits. Well, one of the things that we're really interested in doing is not just doing a whole genome kind of level analysis, which is what we did before, but maybe scanning the genome chromosome by chromosome, and then asking, can we pinpoint those loci that are the specific loci that are contributing to these genetic correlations. And so this was a collaboration, again, with our Michigan State colleagues, and we basically kind of applied a similar methodology, but used windows of the genome. So we would define the windows with sets of genetic markers that were in local linkage disequilibrium. And so then we could compute in each of these windows genetic parameter estimates and see if we could get any significant hits, just like you would a GWAS, but it's like a GWAS of pairs of traits. So what we know is that the phenotypic correlation of urate and kidney function is negative. So I kind of mentioned that before. So if you have bad kidneys, that is your EGFR, that's the glomerular filtration rate. If that's low, you have chronic kidney disease, you're not filtering urate very well, so you have high serum urate. So the overall phenotypic correlation of urate and EGFR is negative. The overall genomic correlation is also negative, and the question is, can we look at specific genes across the genome and ask whether or not the genetic covariance estimates in those local regions are associated with chronic kidney disease or EGFR and serum urate. So you might predict that if you find a significant local genetic correlation, that it would be in the same direction as the overall genomic genetic correlation. So that's kind of what we did here. And again, just kind of trying to get at genes that would be interesting to study for gout. And so here we go. This was the main results from our study, and what's shown here are the genetic parameter estimates for EGFR and urate for a number of different, for 25 different genes. So that none of these had, the Bayesian kind of prediction intervals that overlapped zero, or posterior intervals that overlap zero. So, again, we might have expected them all to be negative, but in fact, there's a solid handful of genes that exhibit the opposite pattern. So these are genes that are whose genetic effects increase urate and GFR, or kidney function. That's kind of cool, I think. We're interested in that, and have we followed up on any of these? No, but I'll just go ahead and kind of tell you what they are. One of the striking things from this study was that the large effect urate transporters that are expressed in the kidney and elsewhere as well, and there are also large effect gout genes too, but acting through the urate pathway to gout. Or ABCG2 and SLC2A9, they didn't exhibit any genetic covariance with urate and EGFR. That was a little bit surprising to me. I wouldn't have expected that. And then, of course, there were a number of loci that handful that exhibited that odd result that the genetic covariance was contributing simultaneously to increased kidney function and urate levels, when we kind of would expect the opposite based on the genomic genetic correlation. So some examples of those were A1CF, LRP2, and GCKR. So A1CF is important for RNA editing. LRP2 is kind of involved in lipid metabolism, and then GCKR is I would call it this gene is involved in a lot of different traits. It's almost promiscuous in its association with multiple different traits. It's highly pleiotropic. Did y'all hear that? Okay, we'll have to edit that from the transcript. So I'm kind of switching gears a little bit at this point. I just want to tell you about kind of a fun study where this was a project with Tony Merriman and our student Nick Sumter. But we were kind of interested in what can we estimate the genetic risk for gout severity in multiple different cohorts, and this study involved genotypes and a genetic risk score. So the genetic risk score was for gout, and then we applied this genetic risk score, which is just the accumulation of genotypes for multiple different loci, and then ask if the sum of the genotypes across those gout risk alleles was associated with the severity of gout, and so which was measured by age at onset and number of tophi. A bunch of different data sets and different ethnic groups, including Polynesians, which is an interesting story on its own, and Tony knows a ton about that. So if you want to email him, just send him an email and say, can you tell me about Polynesian genetics and gout? He'd talk your ear off. So the other question we're going to ask is if there is genetic risk for age at onset or tophi, is it does it depend on whether you're male or female? And so this is I'll just kind of try to be brief here, but let's see. These are the results for age at disease onset, and what you notice here is that the gout genetic risk score is associated with reduced age at onset. So, like a 1 standard deviation increase in the genetic risk score is associated with almost a 4-year decrease in when you get gout. But that was only evident in males and not in females. So this was a sex-specific finding, and which was, in my mind, kind of interesting, but maybe not surprising given the fact that females don't generally start to get gout until after menopause, at a later in age. Okay, so we're starting to get into genetics, and we have some findings on pleiotropy and kidney disease and gout, and the fact that we have some pretty good genetic signal for gout disease activity really kind of led us to propose to look at transcriptomics in gout disease activity. And one of the reasons we're really interested in transcriptomics is that the cellular response to gout-specific signals could be manifested in differential expression of genes that are associated with gout and gout disease activity. And so it was kind of like one step closer, from genotypic information. We have this we have readouts of mRNA and untranscribed or untranslated as well, untranslated reads, and are these associated with gout? Do they implicate any any known or unknown mechanisms of gout flares? So, one other thing I should mention before I kind of move on, I just want to point out that if you're really interested in if you want to read more about the genetics of gout, Tony's research group has published and is actively involved in doing genome-wide association studies of gout, and they've done a lot of work in that area, and it's really it's interesting in its own right. I just didn't explore that or didn't kind of present that in this format just out of time. And I wanted to talk to you guys about the transcriptomic work we're doing. This is unpublished at this time, but we're currently preparing manuscripts for publication on this work I'm about to show you. So this is one of the pathways that's known to cause the acute gout flare. It's called I mentioned it earlier, but it's the NLRP3 inflammasome. And there's kind of two shades of color here. This kind of, is that rose or red? And then on the right is blue. But really, the inflammasome is kind of a two-stage priming kind of mechanism. So there's some signal that interacts with toll-like receptors. It could be bioactive IL-1 beta, a cytokine, and really potent cytokine. It could be TNF. It could be bacterial epitopes that interact with another toll-like receptor. But these are going to kind of jack up the transcription of genes that are going to be important for mounting an immune response to the particular pathogen or sterile pathogen or sterile stimulus. Sorry. And then there's a second step in activation of there needs to be a second signal in the formation of this NLRP3 inflammasome. So once that second signal is realized, there's a formation of this large protein complex that cleaves caspase-1, and then caspase-1 cleaves pro-IL-1 beta, which then is released into the secreted outside of the cell, and then that's the IL-1 beta is the extremely kind of potent cytokine that will initiate the flare. So in a lot of the transcriptomic studies we're kind of interested in seeing signals from this particular pathway. One of the studies that we've done, so maybe not first, but one of the studies that we've done with regard to transcriptomics is to collect RNA packaged gene tubes from participants in a clinical trial called Stop Gout. And so this particular trial was a three-phase trial where everyone in the trial was given urate-lowering medication. So these are the xanthine oxidase inhibitors. And they were dose titrated to less than 7 milligrams per deciliter. So they were titrated to like 6 milligrams per deciliter. And that was very successful. And then the dosage, to do that, they have to increase the dosage of the medications until they can get to this particular level. They then held that dosage constant for another 24 weeks. That's phase 2, or the maintenance phase. And then in the third phase of the trial, the number of flares were counted. And so we actually collected these RNA packaged gene tubes at 24 and 48 weeks, and we were wondering whether or not we could predict flares with these with information on gene expression. So how many did we have? Well, we collected 65 samples at both 24 and 48 weeks, and then we had some unique individuals where we didn't have paired samples, so at 24 or 48 weeks. But we have a longitudinal design and on a rather large data set. So it was over, let's see, I think it was near 300 samples of RNA. So that RNA was extracted from the tubes and then sequenced up at Hudson Alpha, and then they give us the the FASTQ files, and we aligned them to the transcriptome, and then had counts of 20,000 or so genes for each of these 300 people in the study, and then asked whether they were associated with flares. And so the flares were frequent, which was kind of surprising to me, because they were lowering urate levels, to way less than the crystallization threshold, but the frequency of flare was near 50% in the flare observation period. So I kind of already indicated that. So we just used sort of standard methodology, although we did do something a little bit different. We had a mixed effects model where we just accounted for repeated measures with a subject-specific intercept. So this is a mixed model where the fixed effects are week, 24, or 48, flare, yes or no, and their interaction, and then their effects on gene expression. So let's see. This was these are the basic results here. So we had 155 main effect genes that involved time, and those are shown in the upper right circle. And then 113 were associated with flares. Okay, so that's a main effective flare. And then there were 206 that were significant interaction effects. So what that simply means is that the effect of the effect observed for flare depended on whether you were 24 or 28 weeks. Okay. So this is a volcano plot, which is often you see. So a nice way to summarize the results from a gene expression study, which involves a lot of information. It's a lot of genes. I mentioned we had about 20,000 that we estimated, and this volcano plot will tell you. So on the x-axis you have a log2 fold change, and this is going to tell you. So if it's in the red, that's going to tell you that the expression of the gene was increased in people with flare, and then if it's in the blue, then the expression of the gene is decreased for people that flared. So those are the results. And a couple of things that kind of pop out here is these immunoglobulin genes. That was the first thing that really caught our eye, and it was just kind of like, why would we expect to see a lot of immunoglobulin genes coming out here in the study? Because immunoglobulin genes are expressed in B cells, and those are adaptive immune cells. And like the mechanism of gout flares is typically thought to be due to myeloid lineage cells, not lymphoid lineage cells. So that was the first surprising thing. The other surprising thing was that we got a lot of genes that were underexpressed with flares, and these were I have 4 of them highlighted here, but these are neutrophil degranulation genes, and they're uniformly underexpressed in people that go on to flare. So that was one kind of curiosity, I would say, with this data set, the results of this analysis of this data set. So here are the overexpressed genes, and again, I've highlighted those immunoglobulin genes that are kind of the curiosities in this particular study. So what do we have here? So we have a lot of times, another thing that people like to do is throw these genes in this gene list of sort of, significant genes into a pathway analysis, and what we got was really pathways that were significant were due solely to the prevalence of these immunoglobulin genes. So we got things like classical antibody-mediated complement, Fc gamma receptor activation, complement. So there were basically the pathways were dominated by the increased expression of immunoglobulin genes in the people that went on to flare. So what do we conclude from this? We can identify specific genes that are associated with future gout flares, and I think that's the kind of that's an important thing to say. We weren't studying the genes that were associated with acute inflammation. We were collecting RNA at some time t, and then estimating its association with flares that were observed in some t plus t naught time. So there was no direct links with the NLRP3 inflammasome, and that may be because we weren't studying the gene expression in these people that are actively flaring. But I think one of the things that I think that what we're kind of observing here is maybe these genes are sort of telling us about a heightened state of inflammation that's predictive of future gout flares, and the immunoglobulin gene results are curious, and I think it may suggest that there is a role for the adaptive immune system in future gout attacks, but that really we have not validated this yet, so that we can't get too excited about that. Although it's kind of it's definitely provocative. And then finally, the reduced expression of neutrophil granule proteins is also associated with future flares, and this is another thing that's kind of hard to that's sort of hard to interpret. One of the things that it may be telling us is that the reduced expression may be a kind of a marker of heightened neutrophil activation. So these may be neutrophils that are sort of poised to initiate a gout attack. That's possible, but we have a lot of work to do to sort of validate some of these findings. So here are two genes that, again, you can put in your pocket and take to your cocktail parties. We have a immunoglobulin gene. There's one of four, and then this CAMP gene. So this is cathelicidin antimicrobial protein peptide, and there are also three other neutrophil granule protein genes that were significantly underexpressed in people that went on to flare. So I think this would be a good time to kind of move on and say thank you, and particularly thank you to just my colleagues and here too, Ryan and Haymont, but Tony and Jeff Edberg and I, a lot the transcriptomic work was funded by a R01 that we got several years back. And so thank you for inviting me, and I would just be delighted to address any questions that may come up or clarifications. So thank you.